Okay, so today I would like to discuss the FPLC process. So today we'll discuss protein overexpression. But first, I would like to discuss what is MGH. The M in MGH stands for malate dehydrogenase. The G in MGH stands for green fluorescent protein, and the H in MGH stands for the 6-histidine tag. For their overexpression, I started with a well-isolated colony on a freshly streaked plate. It is grown in liquid media to an optical density of 600. This creates an uninduced cell culture. Then the cell line was obtained as a frozen glycerol stock. Using a sterilized loop, some of the frozen glycerol stock was transferred from the top of the tube and spread on the LB agar plate with ampicillin. It is then cultured overnight for 10 to 12 hours in the incubator at 37 degrees Celsius because this is the optimal temperature for E. coli growth. Then the chemical IPTG was added. IPTG is the chemical signal used to overexpress the protein. This creates an induced cell culture that is placed at a temperature of about 20 degrees Celsius to slow the growth and activity of the bacteria. The bacteria are stored at a low temperature to prevent the production of inclusion bodies, which is when the protein recognizes the production of a foreign protein and attempts to precipitate it to remove it from the solution.
Cleared lysate preparation. So we'll get into this in more detail as we go through the steps, but I just wanted to put in for a list of the buffers that we were used for this cleared lysate. Buffer P1 contains RNase A, which will degrade all of the RNA found in the solution. Buffer P2 contains sodium dodecyl sulfate, or SDS, and sodium hydroxide. This will turn the solution a blue color, and once it turns blue, we know that our DNA and proteins have been denatured. And finally, we had buffer N3, which is acidic potassium acetate, which has a high potassium ion concentration, which clears the SDS and the NaOH. So the steps for the preparation of the cleared lysate. Once the cell pellet has been prepared, next we're going to do resuspend, lyse, neutralize, and separate. And as we go through these 4 steps, I want to keep in the back of your mind what is in the solution. At the beginning, we have RNA, DNA, proteins that we don't want, and our MGH protein. Step 1, resuspend. Whenever we resuspend, we place the pelleted bacterial cells in 250 microliters of buffer P1. Remember, buffer P1 is the one that will degrade all of the RNA. Once we add this buffer in, we're going to transfer to a microcentrifuge tube and vortex the solution to mix thoroughly. Make sure that there are no clumps. This indicates that everything has been thoroughly mixed. So, since buffer P1 was added, we can get rid of our RNA. Next, we have to lyse the solution. So once the cells are resuspended, we're going to add 250 microliters of our buffer P2. We're going to invert about 4 to 6 times, and once it is the blue color that I had previously mentioned, you know that it is mixed thoroughly. Remember that this contains the SDS and the sodium hydroxide, so this is going to denature our DNA and our proteins that we do not want. But unfortunately, it now adds the SDS and sodium hydroxide to our solution. After we lyse, we're going to neutralize the solution by adding 350 microliters of our buffer N3, and we're going to mix. Initially, the solution is cloudy, and the pH of the solution was changed, but soon after the solution became colorless again. And after this N3 has been added, the SDS and sodium hydroxide have been removed, leaving only our MGH protein, theoretically. Finally, we want to separate the remaining solution by centrifuging at 13,000 rounds per minute for 10 minutes. All of the liquid portion is considered our final cleared lysate, ready to be separated via FPLC. What could we do differently? So group 2 performed a cleared lysate dialysis. And what this means was we took 10 milliliters of this cleared lysate, and we took 1 liter of a buffer, and we put them in a dialysis bag that has a cutoff of about 30,000 molecular weight. We let it sit for 24 hours, and what was left was what group 2 used.
Now we'll discuss the FPLC process. I will also discuss the important parts for purification. This instrument is similar to doing an automated version of column chromatography. The process starts by having buffer A from one flask and buffer B from another flask, both leading into the mixer. The mixer mixes the buffer A and B together. Then it goes to a peristaltic pump. The purpose of this pump is to move the mobile phase, which is the components of the buffer. The next part of this process is the sample injector. Then the next stop is the column, which is the stationary phase. In this part, there are different options to use for the column material, which is known as the resin or beads. The next part is the UV detector, which is a little spectrometer that measures two wavelengths. It measures 260 and 280. 280 measures protein and 260 measures the nucleic acid concentration for DNA and RNA. Next part of the process is the conductivity meter, which measures ion concentration. This will show how salty the solution is. The saltiness can be due to the components such as salts, proteins, and nucleic acids that are charged. The next part is the diverter. It can divert to waste if we are not interested in saving what is coming through, or we can go to the fraction collector. The diverter can communicate with the UV detector and the conductivity meter. This allows for it to determine whether to move the solution to waste if there are no proteins coming through, or to the fraction collector if proteins start coming through. The process just described is considered the main line because it is a non-stop and there are no passengers that are picked up. The other pathway is the bus stop, which stops at the sample loop to pick up our passengers, which is our protein. This is what the sample injector is for in order to load our column. Thus, this is where our protein will be loaded and the knob will be turned to offshoot. Then after the bus stop picks up our passengers, it goes back down to the main line and it is going to go to the column to continue on the original pathway.
So in the FPLC, we have to use those lysis buffers as previously mentioned. Buffer A and B both contain very similar things. They both were 500 milliliters total in volume and contained 50 micromolar concentration of phosphate with pH 8 buffer, 300 micromolar sodium chloride, 0.1 micromolar PMSF, and 1 micromolar DIT. What differs between buffer A and buffer B is that buffer A contained 10 micromolar of imidazole and buffer B contained 250 micromolar.
For our column, we used a 5 milliliter nickel NTA column. It was prepared from fresh resin equilibrated with 10 volumes of buffer A at 1 milliliter per minute. For the FPLC run, the minutes of 1 through 30 were at 1 milliliter per minute with 100% buffer A. For minutes 30 through 75 at 1 milliliter per minute and it had a gradual increase of buffer B from 0% to 100%. The fractions were collected every 3 minutes with a volume of 3 milliliters. Overall, the total fractions we had were 25. So then I will discuss the FPLC data and analysis. For the first group, there was no clear lysate dialysis. So for our first data graph on the FPLC traces, the blue line represents the protein concentration and the red line represents the salt concentration. The red line also shows the conductivity of the sample, which refers to the number of ions in the solution. The x-axis shows each fraction taken. The blue peaks shown were which proteins were possibly there. The dip in the red line shows where the cleared lysate went through, which is why it had a low salt conductivity right there. So for the next slide, I would like to highlight the protein of interest, which is MGH. The second peak determines what fractions I will want to pull due to the presence of MGH here. For this data, I would want to pull fractions 9 through about 25. It can be inferred that early fractions likely contain contaminants. However, this cannot be confirmed until work from the other teams has been done.
Group 2 was the one that had the cleared lysate dialysis. Our results showed similar things where we had the blue being the protein and the red being the salt. However, my region of interest did not have a high as high of a peak as the other group, but the fractions were similar. Mine was 18 through 23.
So in conclusion, it appears that it's necessary to pool fractions 19 to 25 because the MGH protein is located at the second peak, and this data was for the first set. Overall, the FPLC is a good technique to find the general location of the protein of interest. However, further analysis from other teams must be performed to identify the exact fractions that do not contain any contaminants.
I had very similar results. Overall, the cleared lysate dialysis seemed to have played a part in the FPLC trace. Um, Once again, we wanted to pool fractions 18 through 23 to undergo further analysis from other teams.